Thank you for joining us for our Great Plains Fire Science Exchange webinar. I'm Sherry Leese. I'm the program coordinator for the Great Plains Fire Science Exchange, and I'll be moderating the presentation today. A quick note about the exchange. We are dedicated to making fire science accessible in the region, and our focus is on grassland systems of the Great Plains. Let's go ahead to the next slide. I do want to share with you some of our upcoming events. Next week, we are participating in a fall burning field day at the Tallgrass Prairie National Preserve in Strong City, Kansas. We'll also be out at the Association for Fire Ecology Congress in San Antonio next month. We have some upcoming webinars scheduled. December 1st, Casey Olson will be sharing his research on Sericea lespedeza and growing season burns. We have a presentation coming up that will look at juniper pollen and its effects on health. And then we will also be out at the 6th National Grazing Lands Conference in Grapevine, Texas. And lastly, we are working on getting Derek Scasta to talk about the role of fire in controlling hornflies and ticks in grasslands. So if you're not familiar with the exchange, please visit our website at gpfirescience.org. You can also find us on Facebook and Twitter, and we have numerous presentations on our YouTube site as well. So for those of you not familiar with the Adobe Connect environment that we're using right now, the participants will be muted, but you can write your questions and comments into the chat box at any time, and we will work on answering those towards the end of the presentation. You can also let us know if we need to speak more loudly or slowly or things of that nature. There's a little icon of a man or a person raising their hand, and under that drop down, there's some ways to interact with us there. So I want to introduce our speaker for today, Alice Tipton. Alice is originally from St. Louis, Missouri. She has a bachelor's degree from Truman State University and is currently a PhD candidate with the University of Missouri in the Biological Science Department. Her work focuses on plant interactions with soil microbial communities. She's looking at what biotic and abiotic factors drive these relationships and how soil inoculation can be used in plant community restoration. She's worked extensively with education, and as a National Science Foundation GK12 fellow, she has also focused on science outreach with elementary school students and teachers. So Allison, go ahead and unmute your microphone and take it away.
Okay, great. Can you everybody hear me okay? Could somebody maybe write in the chat box that they can hear me so that I know that I'm coming through? Okay, I see somebody's typing. Great, thank you. Thank you so much for being here today, and thank you, Sherry, so much for inviting me to talk. I'm going to talk a little bit about some of my PhD work that I've been working on over the last 5 or almost 6 years now. And just to kind of outline what I'll be talking about today, I'm going to give a general introduction to what mycorrhizal interactions specifically are and think about them more in the terms of grasslands where I work and where most of us probably spend a lot of our time and think a lot about those systems. I'm going to then talk about a project that actually has nothing specifically to do with fire, but takes place in a habitat that is extremely fire dependent. Some work I did on looking at how mycorrhizal interactions are affected by edge effect, so cross core and edge habitat in these glade systems that I work in. And then lastly, just because, mostly because this is a fire exchange, though I don't specifically study how fire affects mycorrhizal interactions, at the end of the talk today, I'm gonna go into some details about some work that other people have done and kind of discuss what we know about how fire and prescribed burns affect interactions between mycorrhizal fungus and fungi and plants. So to get started, this is the habitat that I spend most of my time in. This is an area called a glade, and these are located on the south and southwestern facing slopes of the Missouri and other Arkansas as well, Ozarks. And they're these dry, rocky grasslands that with persistent fire moving through the system, and we know that fire was extremely important to the diversity of plants in the Ozarks historically. With persistent fire, these remain as grasslands and remain very open. Um, but due to fire suppression over the last 200 years, many of the glades now look like an eastern red cedar forest. So we know that fire is really important to keeping these glades open, and they contain species of both plants and animals that we really don't find other places in Missouri. So for example, we have collared lizards that live in glades. We also have a couple plant species, specifically a plant Echinacea paradoxa, which we really only find in glades. So they're very important to the general biodiversity of the Ozarks. And what I think about when I'm working in one of these areas, these glades that are these open grasslands, diverse plant community, almost completely surrounded by this woodland of oak and hickory trees, I'm really thinking about what's going on underground. So all of these plants are interacting with microbes that we can't see that have the potential to have huge effects on how these plants are surviving, how they're interacting with each other, and the community composition within these systems. And the main type of interaction between plant and soil microbes that I spend most of my time thinking about is their interaction we call mycorrhizae. And this is a relationship between the plants and a fungus that lives in and on its root system called mycorrhizal fungi. And it's a symbiotic relationship between the plant and the fungus where the fungus is able to uptake things like phosphorus, nitrogen, and sometimes even water that the plant can't obtain on its own in, in exchange for some of the plant's carbon or sugar resources. And this is usually considered a mutualism. We do know that sometimes it can sway more on the pathogenic side. We usually think of this as a mutualism. And over 80% of all land plant species on Earth make some type of mycorrhizal relationship. And we know that we believe that plants would not have actually transitioned to land evolutionarily without these mycorrhizal fungi. And of those 80% that make some sort of mycorrhizal relationship, 80% of those make relationships with a specific type of mycorrhizal fungus that I study that I'm going to talk about today. This is one of my favorite pictures, really showing the effect that mycorrhizal fungi can have on plants. This is a picture of a pine seedling that is interacting with a ectomycorrhizal fungus. And if you look here, these brown lines here are actually the pine's roots. And all of this white material is hyphae from the ectomycorrhizal fungi that's reaching out into the soil. So not only can the fungus potentially change the forms of nutrients, excuse me, to make them more available to the plant, but it also can really drastically increase the surface area over which that plant can uptake resources. And we have a professor here at the University of Missouri, Gene Mahale, who likes to always say that James Cameron must have known something about mycorrhizae when he was thinking about Avatar, because it kind of is has the potential to be like what we think about when we watch this movie and thinking about Pandora, where these plants can almost be connected to each other in some way because of this mycorrhizal network and interaction.
Let's see if we can get her back.
The two main types of mycorrhizal fungus that are most common on Earth are this type ectomycorrhizal fungi, which has been greatly studied because it's easier to see. These are a lot of the mushrooms that you think of when you're walking through the forest. And they tend to associate specifically with plants like oaks, hickory trees, and pine, for example, among many others. But the most common type of mycorrhizal fungus is the one that I study and the one that is most common in grasslands, which is arbuscular mycorrhizal fungi, or what I'm going to call AMF throughout this talk. And AMF gets its name from, if you look at this picture down here, this is called an arbuscule, which forms inside of the root cell. And this is finely branched hyphae that is the location where the exchange between the plant and the fungus takes place. And so that's how they get their name from these structures called arbuscules. And a lot of the work that I do, we calculate mycorrhizal interactions through mycorrhizal colonization of the root system. And to do this, we clear the roots with chemicals and then sting the fungus specifically with a chemical called trypan blue. And so all of this blue material here is the mycorrhizal, the AMF. And this is a picture that I took from some of the work I'm going to talk about today. And these square-like structures are the root cells. So you can see that that's how we determine if mycorrhizal fungi is there, by looking for that stained blue hyphae and also those arbuscules, which we see here. And we know that in grasslands, these interactions between plants and AMF can be important drivers of things like community composition um, and competition between plant species, as well as plant community diversity. And also they can have drastic effects on productivity, and it depends on which plant species we're talking about. So some plant species and plant taxa tend to be more reliant on AMF than others, but this is a picture here of Panicum virgatum, or switchgrass, taken by Gail Wilson. These plants are the same age. This is a switchgrass growing without its mycorrhizal partners or its AMF partners, and this is the same species growing with mycorrhizal partners. You can see here that can have huge effects on plant productivity. And we also know that in regards to this relationship, the AMF rely on a plant host in order to survive. They cannot survive without a plant host. And it's thought that the plant itself can survive without the fungus, but you can see here that oftentimes productivity and other things are largely affected when that fungus is not present. And so when I started to work in these glade systems, these kind of isolated grasslands scattered across south and southwestern facing slopes of the Ozarks, we were kind of interested in how mycorrhizal interactions would be specifically affected by certain patch quality characteristics. So as I mentioned, a lot of these glades fill in with cedars during fire suppression, but many of these glades are currently being restored, which means that they remove this eastern red cedar trees and return fire to the system. And so we had glades that varied in restoration age. We also had glades that varied in size, how isolated they were from other glades. But then we also were really interested in this idea of looking at the difference between this core glade habitat, so mycorrhizal interactions in these grassland species in the core habitat versus near this woodland transition zone or the edge. So in this initial study that I did where I looked at mycorrhizal colonization within the plant roots of the glade plant community across these sites, one thing we found, and this graph here is representing, so the proportion of root length of the plant community colonized by AMF is on the y-axis, and then the restoration age of the glade is on the x-axis. And one of the only characteristics that was as an indicator of mycorrhizal colonization across sites was restoration age. So as glades had been opened for much longer, as cedars had been cleared and fire had been returned, mycorrhizal colonization increased within the roots of these glade plants. But as I mentioned, we were really interested in this edge effect. So we also examined how mycorrhizal colonization varied between that core central habitat and that edge woodland transition zone. And a couple reasons why we thought this might be important was, well, first of all, I mentioned that oaks and hickories tend to associate with ectomycorrhizal fungi, so there could be some interactions going on in the soil between these two different types of mycorrhizae. We also know that the conditions, both abiotic and biotic, could be different between this core and edge habitat, and so we were interested in that. And what we found was that the difference between core and edge was a better predictor of mycorrhizal colonization within a site than some of those across-site variables like size and isolation. So this graph here on the left, the green bar represents core habitat, the orange bar represents edge habitat. These are samples that were collected randomly from plants throughout the glade community, and we saw that in the core habitat, mycorrhizal colonization was higher. Then we also looked at mycorrhizal colonization in two specific plant species that we could find in almost all of our sites growing in both the core and the edge habitat. So we looked at mycorrhizal colonization within the roots of these. One is Rudbeckia missouriensis, or Missouri brown-eyed Susan, and then another is Schizocarium scoparium, or little bluestem. And both of these plants, the same species, plants growing near the edge, showed lower colonization, AMF colonization, than plants growing toward in the core habitat. And so that got us asking, well, what causes this variation in mycorrhizal colonization in the edge versus the core habitat? And that's the study I'm going to talk about today. As a nurse, is a study in a common garden experiment that I conducted with two elementary school teachers. So Sherry mentioned that I work closely with elementary schools. These are both teachers that were working with me in their classroom. Oh, I'm sorry, it sounds like somebody lost me. Okay, so that seems like it might just be Amy. So both of these teachers, I worked with them in their fourth grade classroom during the school year. And then my advisor, Candace Galen, thought it would be really interesting to see if they would want to do their own research project out in the field the summer after I'd been working with them in the classroom. And they were very enthusiastic. So the three of us put together a research project, and they were my field techs for the summer, and then also conducted the experiment I'm going to talk about today. And it was really fun working with them because they're very different. So Carla, picture on the left, was a horticulture major in college, and so she was really excited about being outside and digging within these glades. And Becky, on the other hand, was a little bit more hesitant, but she wanted to get the experience. And I love this picture of Becky because about she said she didn't want to use any bug spray while we were out working that summer, and this is about 5 minutes after we arrived at our first site. So she changed her tune very quickly when she experienced her first tick. And so the ideas we were wanting to explore with Carla and Becky's project were what kind of things are causing this variation between mycorrhizal colonization between the edge and the core habitat. And one, as I mentioned, is environment. So we know it's shadier near the edge due to this canopy cover. There might be some differences in soil nutrient levels. And so there could be some environmental variation that's causing this. Um, we know that, as I mentioned before, the edge habitat has tree species that associate with ectomycorrhizal fungi. There could be other microbes in the soil that are affecting this relationship. So we thought maybe the microbial community in the soil could be different between the core and the edge. Maybe there's some differences in physical soil properties. We know there's a lot more leaf litter near the edge. Maybe there's some of these are causing this variation. And then lastly, we had this idea that what if the plants themselves were different? So we had two plant species. The same species was doing different things at the core and the edge. So we thought, could there be something different about plants that are growing near the edge of a glade versus plants that are growing to the core? So could there be this edge ecotype? And this is a lot to explore, and all of these variables could be interacting in many different ways. So what we decided to do was do a common garden experiment and eliminate our environmental variables like shade from between the edge and the core, and really focus on the microbial community, potentially physical soil properties, and this idea of plant ecotype. And the main questions we were interested in asking were, one, do core soils provide a larger mycorrhizal potential than edge soils? So for example, are these plants showing higher colonization because there's just more mycorrhizae there for them to associate with? We had this question is, are we seeing higher colonization in these core plants because the core plants themselves are more responsive to soil inoculum? And this is in regards to both higher colonization, but also we didn't look at growth for any of these. So we were just assuming that higher colonization meant better growth for the plant. So we wanted to look at that as well. And then lastly, do core and edge plant ecotypes show local adaptations to microbial communities? So for example, do core ecotypes grow better in core soil, and do edge ecotypes grow better in edge soil? So is there some kind of local adaptation within sites? And so to address this question, and this is a map of the fire exchange regions, I am working in that little yellow box here is where we did this work. So not actually in the Great Plains area, but kind of in the oak woodlands of the Ozarks. So if we zoom in on Missouri, in that black box there. And then Carla and Becky went out with me to 3 different glade sites that I had worked in previously. And they collected both little bluestem plants, so adult plants, and the soil within the rhizosphere in 3 locations, approximately 4 meters into the glade from that edge, so our glade edge area, and then 3 locations within the core of the glade. And what's great about working with a grass like little bluestem is they were able to take these large clumps of little bluestem that they collected. We took the soil from that, we saved that, and I'll talk about that later. But they were able to use that little bluestem to almost clone it in a way. So you can separate the ramets by using a razor, and then you can actually remove the majority of that root system, put the shoots into cups with distilled water, and then those shoots will grow back their own individual fresh root system. So we are able to clone edge ecotype and core ecotype plants and grow them back fresh, sterile root systems, which was a great system to work with for a question like this. And then those are the plants that they used in their experiment. This is a picture of Carla and Becky setting up everything and getting ready to move it outside to our common garden location. So they used those core ecotypes and edge ecotypes, and we set up a classic home and away experiment where we were able to plant core ecotypes in pots that contained sterile, homogenized soil for most of it, but had a thin layer of core soil. So this brown bar here represents where we put our inoculum. So this is the soil that we collected with core plant rhizospheres from all 3 glades. We mixed that all together to create our core inoculum, and that's located here. So we were able to plant them in plots that were inoculated with core soil, put core plants in pots that were inoculated with core sterile soil, and then also in the away situation. So core plants in edge soil, core plants and edge sterilized soil. And then we could do the same thing with the edge plants. So we have this classic home and away experiment where we have these plants growing in both their home and away soil with inoculum and then sterilized. They let these plants grow for approximately 3 months. This is the first time we'd ever done this cloning experiment, so we had higher mortality than we were originally thinking. So about 65% of the plants survived with an N of about 43, and the sample size for each of those treatments is listed below the plot. Okay, so even with this kind of small sample size, we saw some interesting results. So this first graph here is the number of new leaves shoots grew in the different treatments. Core and edge soil did not show any variation in growth. So these bars here on the left represent the core ecotype plant growth, and on the right represent the edge ecotype. And this is core plants grown in core inoculated soil, so core or edge inoculated soil, and then core plants grown in sterile soil, and vice versa, so edge ecotype and inoculum, and edge and sterile. And so the green bars here represent core ecotype, orange bars represent edge ecotype, and hashed represents sterile treatment. So the first thing that we saw, and this was not too surprising, is that plants grow better when they have the microbial community present. And this microbial community included Mycorrhizae and all other microbes that were present in the soil. So plants grow better when they have this microbial community present, and that's pretty common knowledge for little blue stem. But what was really surprising to us was we had this plant ecotype by inoculation interaction. So both core and edge ecotypes grew better when they were inoculated with a microbial community, but core plants did 58% better than sterile controls, and edge ecotypes did 33% better than sterile controls. And what you'll notice is the main reason why we see this difference is because these core plants did much worse in terms of growth than edge plants did in sterile conditions. And so this suggested to us that, and this really surprised me, I really didn't think that we would see this, that there is this possibility that core plants are more reliant on their microbial community for growth. So this next graph is the proportion of root length colonized by AMF, so kind of similar to the data I was showing you before. And we have core ecotype on the left here and edge ecotype on the right here. And in this graph here, a darker bar represents an ecotype in its home soil, and a lighter bar represents an ecotype in its away soil. The sterile controls are not included in this graph because we saw anywhere from 0 to 5% colonization within our sterile controls, which means they worked. We didn't have much, if any, mycorrhizal fungi or AMF in our sterile controls. And so what we found in this graph here that you'll notice is this soil type by plant ecotype interaction for mycorrhizal colonization, where for core plants, they showed higher mycorrhizal colonization when they were growing in their home soil as opposed to edge soil. And edge plants did show higher colonization in their home soil, but it wasn't significantly different from when they were growing in the away soil because their colonization rate, which in the edge soil is much more variable. So what this suggested to us, again, was that potentially there's something going on differently in these core ecotype plants as opposed to these edge ecotype plants that is dictating mycorrhizal colonization. And it also was true that edge soil was much more variable in general for mycorrhizal colonization than center or core soil. And that's not too surprising to us because if you think about how the edge looks, it's a much more variable environment. So that there's that potential that this mycorrhizal community potentially in edge soil is much more variable than what we see in core soil. And so to look back at our questions, our first question was, do core soils provide a larger mycorrhizal potential than edge soils? And our results would say no. We didn't see that all plants showed higher colonization when they were in core soils than edge soils. They seem to be getting colonized in similar ways depending on which soil they're in. But we did see that the edge soil caused more variability in mycorrhizal colonization. Our next question, are core plants more responsive to soil inoculum? And our answer here is yes. Core plants do worse in sterile soil than edge plants. So they are more reliant potentially on this microbial community for growth than edge ecotype plants. And lastly, do core and edge plant ecotypes respond differently to home away soils? And our answer to this question based on our study would be that growth response was not different. So core plants didn't grow better in core soil. Edge plants didn't grow better in edge soil. But we did see that core ecotypes showed higher AMF colonization in their home soil. And so this again suggests that something different is going on with this core ecotype as opposed to this edge ecotype for little blue stem. So just to kind of recap, this little blue stem shows this potential for local adaptation to edge and core microbial communities, and that these ecotypes interact differently with the soil community and with AMF. And one place we'd like to take this in the future is that we were using this plant, little blue stem, that grows in prairies, grows in glades. It's a pretty general endemic, like this plant shown here on the right bottom, Echinacea paradoxa, which is more endemic to these glade systems. And lastly, I just want to mention that I think studying a naturally fragmented grassland like these glades here can really teach us some things about core and edge effect, specifically with in terms of mycorrhizal interactions. When we're thinking about grasslands that are more fragmented than they once were. So this is a picture of one of my favorite prairies here in Missouri, Schwartz Prairie, and you can see the row of trees along the back. So these places like prairies that maybe have more forest edge than they once had. I'm thinking about how these plants are interacting with the soil community in the center of these prairie fragments as opposed to near that edge might be an important thing to think about in the future. Because this is how our landscape looks like now. So I think thinking about edge effect could be important when we're thinking about restoration of grasslands. So with the end of the first part of the talk, real quick, I don't have much time left because of the little the part where I left for a second, but I just want to thank all the people that helped us with this project and the funding sources as well. So I'm going to switch gears a little bit here. I've talked about edge effect. Now I'm going to switch to something a little bit different and talk about something that I haven't specifically studied in detail, which is fire and mycorrhizal interactions. And I have this little warning here. As I mentioned, I haven't studied this specifically before, so I am not a fire ecologist. This is a picture of me on my only burn I've ever been on. And as you can see, I'm excited and looking pretty goofy and naive. So I work in a system where fire is extremely important. but this is not something that I've studied in particular. So what I'm going to do is talk a little bit about the ways in which we think fire could affect AMF, and then show you a few studies that looked at this in more detail. So when I was thinking about, I think about this question a lot, how is burning impacting these interactions that I study? And I can think about the ways in which fire could affect this in 3 different ways. And first of all, I want to mention I forgot to put a photo credit. This picture here on the top was taken by Benjamin Zach of a burn at Tucker Prairie here close to the University of Missouri. So one way that we can think about that I've been asked a lot about is this idea of soil sterilization and temperature affecting the fungus. And so I'm going to show one study at the end that kind of talks about that in more detail in regards to slash piles and burning. Um, and then the other 2 ways that I think about it are, I think that fire, so this one on the left, the soil sterilization, I would consider more of a direct effect. The fire is heating up the soil and causing potential changes in the mycorrhizal community. I think that indirectly through the plant community is another way that this could really be important. So as I mentioned, these AMF are almost completely reliant on a plant in order to survive. They need that root system to get their sugar. And so they could be indirectly affected by fire through its effects on the plant community. And talk a little bit about that and some of these studies I'm going to mention. And then lastly, through environmental conditions. So after a burn, it's more light hitting the surface of the soil. There's potential release of volatiles of certain nutrients. And so changes in the environment could also potentially affect mycorrhizal interactions. But one thing I want to say specifically, and I think that the studies I will show you will allude to this, is that we think of grasslands, specifically the ones that I work in, as places that are dependent on fire in order to remain open. And these mycorrhizal fungi that are living in and on these plant roots have been have evolved through this fire just as these plants have. And so if they're completely reliant on this plant community in order to survive, one thing that we can think is that frequent burns are most likely not causing any huge detriments to these communities because they've evolved to deal with these situations and these types of disturbances. The papers I'm going to talk about next talk about use a couple different ways to look at AMF, and so I'm going to mention that real quick. I talked about mycorrhizal colonization, which is what I have used in a lot of my work. So how much of the root is colonized by AMF, and that'll come up again. But I showed you that picture of the pine seedling at the beginning interacting with ectomycorrhizal fungi, and in some of these studies, they calculate extra radical hyphae in the soil, which is the hyphae of the mycorrhizal fungus that's in charge of going out and getting resources. And then another tool that people who study mycorrhizal fungi use often is spore identification. So the spores of AMF are very large compared to other types of fungi, and they're very variable in shape and size and color. So oftentimes they can be used as a way to get an idea of diversity of AMF taxa in samples. And so I'll be talking about that a little bit as well. So one of the first study I'm going to talk about was conducted almost over 20 years ago, but I think it's a really important study that looked at the effect of fire on mycorrhizal ecology, and specifically I want to talk about this study because it talks about the same plant that I was just speaking about, which is little bluestem. So what they did here, this took place in a sand prairie in Illinois, and the half of the study site was burned in April of 1984 and half of the site wasn't. And they collected samples multiple times within the first year and then once in the May 2 years after the burn to look at how the soil within both the rhizosphere of little bluestem specifically, but then also randomly outside the rhizosphere in random locations differed in spore abundance due to this burn. They also looked at mycorrhizal colonization of little bluestem across that time period as well. And in the first year, so that first year after the burn, all of the times that they sampled, they saw that there was a decrease in spore abundance in the rhizosphere of little bluestem. So the number of spores decreased within the soil closest to little bluestem root systems. And they also saw that randomly throughout this burn site, there was lower spore abundance compared to the unburned site in that first year. So spores are oftentimes a good way to think about how mycorrhizal communities are changing, though we know that's not the only form which mycorrhizae takes. So sometimes spores can be a little deceptive, but they did see some changes in spore abundance. And then they also, and I think this is important, they also did see some decreases in root AMF colonization within those little bluestem plants in that first year. But in the second year, so it was burned in 1984, so in May of 1985, they still saw lower levels of spores within the rhizosphere of little bluestem, but abundance in the community in general and root colonization of that little bluestem was no longer any different than their unburned site in the second year. So though they did see some differences within the rhizosphere of the little bluestem, they didn't see any major differences between burned and unburned sites within the soil within the whole site, as well as AMF colonization. So what they suggested that this was potentially saying was that was a good indicator that maybe changes within the plant community. So how that fire impacted the little bluestem itself might be causing these differences in spore abundance directly within the rhizosphere of the little bluestem. But I also think it shows that these communities eventually bounce back and that they're adapted to these frequent fires. In another study that was done by Eom et al. In 1999, this was done in Kanza Prairie. So again, I think that's almost 20 years ago, but I think this is also a nice study because what I like about this that's a little bit different than the study we just talked about was that they were working in plots. So they sampled these locations in 1995, and they were working in plots in Kanza Prairie that had been under different burning and mowing treatments for 9 years prior to the collection. And their burn treatment here was that they burned annually in April and May of every year. And then they sampled their sites in August of 1995. And in this study, they looked at a few different things. They looked at hyphal length, so hyphal length in the soil of those extra radical hyphae as well, which is something that the other study did not examine. And what as I mentioned, they were looking at things like mowing and then different nutrient levels as well. But I'm just going to focus on the results for fire. So they collected spores and looked at the diversity of spores to determine AMF diversity of AMF taxa. And they did see that in those sites that were burned annually, they showed lower spore diversity than sites that were not burned. But they saw an increase in spore abundance. So the sites that were burned annually showed lower spore diversity, but higher spore abundance. And this is because most of the different taxa or morphotypes of spores that they examined were actually increasing in abundance in these sites with only a few decreasing. They saw no difference, and this is, I think, a little bit surprising. They didn't see any differences in root AMF colonization between these annually burned and unburned sites. So no differences in the colonization rate within the roots. And they did see some decreases in extra radical AMF hyphae within the soil. And one other thing I want to comment on is that most of the AMF species displayed positive responses in spore abundance to burning, with only one showing a negative response. And so that's why we saw that difference in spore diversity. And I'm going to keep moving because I'm running out of time and I want to make sure I have time for questions. I'm going to actually skip this study because I want to move on to the last one because I think it's really important. But basically what they showed here was that in wildfires, the AMF community changed, and this was in a forest system in China, 1 year after a burn, but returned to similar AMF communities than the control. But I don't have time to go over that, so I'm going to keep skipping and just go to this one because this is something that people have asked me a lot about, is this idea of whether slash piles can cause soil sterilization. And one reason I think I talk about this a lot with land managers is because in the glades where I work, a lot of times they're removing cedar and piling them up and burning them. Sometimes they create a slash pile in the middle of these glades, and I can oftentimes tell where that slash pile's been. So this is a really nice study that looked at how slash piles in in Arizona near Flagstaff in a national forest were impacting mycorrhizal colonization. And so to look at this, they found the remnants of slash piles from a recent burn, and they planted corn seedlings 3 meters into the slash pile area. This was after the pile had been burned. 1.5 meters into the slash pile at the edge, and then 3 meters out. And they used those corn as almost like plant photometers to see how much colonization they showed within their root system in and outside of these slash piles. And what they found was they looked at this directly after the burn, so year 1 and then year 2, and on the y-axis we have AMF root colonization. And you can see that inside of the slash pile area, AMF colonization was much lower than outside. So this suggested that sterilization was occurring in these slash pile locations. And in this study, they also thought it would be good to address seed viability, because if we're thinking that the AMF community is being affected by sterilization, we would think that maybe the seeds would be as well. And so they did a germination study looking at seed viability outside of the slash pile areas, and then both at the edge and 3 meters in, and found that seed viability was much higher outside of that slash pile as opposed to inside. I'm sorry, I'm rushing through this a little bit. But this is, I think, my favorite part of this study, was that the next thing they said was, okay, so if sterilization occurs, what if we amend these slash piles with inoculum soil? Will we see higher percent colonization? So here what they have is they have slash piles locations where they did not add any soil amendment, slash pile locations where they added a sterilized soil amendment control, so very similar to in my study where we had that sterile control, and then a location where they added both seed and soil amendment. And what they saw was when they replanted those corn plants after the soil amendment, they saw a higher percent of root length colonized by Mycorrhizae in slash piles where they amended those locations with fresh soil. And then when they looked at native forb plant cover, they had locations where they, again, did nothing, added just sterile soil to the slash pile, added soil to the slash pile, added just seed by itself, and then added seed and soil. And you can see that when they added seed and soil together, when they included soil with that seed in that second year, they had higher percentage of native forb cover as well as native graminoid cover. So I think this is a really, they show a few examples of how these slash piles might be causing soil sterilization. So with that, I'm just going to end real quick and say that this is complicated, and we know that burns are potentially impacting mycorrhizal fungi, but it really depends on things like fire severity or season, and how these burns are impacting things like the plant community. Soil sterilization um, could happen, but I think it depends on fire severity, and I think that's something we need to keep in mind. So coming from a non-fire ecologist, but somebody who studies mycorrhizal fungi, it's a very variable situation, and has a lot of can have a few minutes to take questions. Is that a good idea, Sherry?
Yes, yeah, we have about 8 minutes for questions. So
Okay.
if you would like to ask Alice to follow up on something that she just described, please write that in the chat box, and we'll go through those as soon as we can see them. So I want to thank Alice while you're thinking of questions for her. I found this really pretty interesting, especially since it's really difficult to get information on what's going on below ground, and it's hard to understand that because we can't see it. So I appreciate
Yeah.
that is what you've decided to work on. It's definitely an important need.
Yeah. Yeah. Thank you. Thank you.
Okay, so it sounds like I don't see anybody typing yet. So
Oh, okay.
let me
Somebody's.
see if I can let me see if I can. So the Joint Fire Science Program had put together a synthesis on fire
Mhm.
effects on soils, and it's interesting
Mhm.
because some of their conclusions were a little bit contrary to some of the things that you had found
Yeah.
in your own search, and
Right. Right.
that illustrates the point that it's pretty complicated, and it might.
Yeah.
I wonder too if it might also be partly a function of what ecosystem types were focused on in that
Sure.
synthesis versus what
Yeah.
you're studying.
Yeah, and when I looked through the literature, and there's always something that you could be missing. But a lot of the reviews that I found that looked at how, well, one review and one of the most recent reviews I found that looked at how fire affects microbial communities, they specifically did not include um, AMF in their analysis
Mm,
because of the situation with its dependence on the plant community. So yeah, that you're right is that there could be some ecosystem situations that might make this different depending on where you are, for sure. Yeah. But I think severe like fires, I think the reason why I've kind of steered away to studying this in my own work, and I think this is why me and fire ecologists need to get together more, is that I have a hard time in my site. So, for example, I tried to look at how long it had been since there was a burn as an effect of mycorrhizal colonization in one of my initial studies, and I didn't see any strong effects of how long it had been a burn. How long it had been since the site had been burned in mycorrhizal colonization. But I had no data on what those burns were like. So, how severe were they? How frequent were they? That was hard for me to get. So I think that for me to learn how to better measure those kinds of things and work together might be a way that we could address some of these questions.
hmm
Okay, I see
Great.
a question from Amy. So she says, tall fescue can reduce the ability of native grasses to develop their relationship with mycorrhizal fungi. How does this happen? That's a really good question. So I don't know as much about tall fescue, but invasive plants can do that in a couple different ways. Some plants produce um, uh, I think would consider it allelopathic chemicals that actually reduce mycorrhizal fungi in the surrounding area and reduce the interaction between mycorrhizal fungi and nearby native plants. And so that an example of that is something like garlic mustard. But plants, though mycorrhizal fungi are fairly general, so the idea is that most plants can interact with most mycorrhizal fungi. There are some differences in the communities of mycorrhizal fungi that live on one plant versus another. So if a plant, for example, a grass, is highly mycorrhizal, it might increase the abundance of mycorrhizal fungi within the soil, but it might only be increasing abundances of fungi that benefit it itself rather than plants nearby. So they can change the community of mycorrhizal fungi sometimes in a way that we think might impact the growth of nearby plant communities or nearby native plants. So that's another way that they can do it. Also, if they're becoming extremely abundant and taking over a habitat and they aren't extremely reliable on mycorrhizal fungi, it's often thought that they can actually decrease the abundance because the majority of the plants are not interacting with them. So there's a couple ways that can happen. That's a really good question. So Casey asks, should we be considering inoculating prairie seedlings, converting from crop to native? That is a good question. And yes, so there's a couple labs that are working on ways to use soil amendments in prairie restoration. So I have another chapter of my dissertation that looked at this for these glades, specifically glades that don't tend to ever come back from during the restoration process. And we looked at how soil inoculation impacted a couple different plant species, and we found that plant species differed and how they responded to that inoculation. But there's some great work coming out of Jim Beaver's lab. Um, and I think if you're interested in that, I would definitely look through some of the literature if you can get your hands on it. And they've done both whole soil inoculations with prairie seedlings, where they use all, the whole soil microbial community. And then they've also isolated specific prairie mycorrhizal fungi or AMF and use those in inoculation processes. And what they find, the general trend of one thing that they find is that later successional plant species tend to show a stronger response to mycorrhizal fungi than earlier successional plant species in these prairies. And that's a really good question.
Alice, if you are interested in sharing some of those resources that you might already have, or websites
Okay.
where people can go and look that up, we'll post it along with the recording that
Okay.
people can
Yeah, that
access
would
on
be
the
great.
website.
Yeah, I could maybe share some of the links to those papers.
Sure, that'd be great.
Yeah.
We
Okay.
have
So,
just about another minute,
okay.
so yeah, if you can answer Pete, and then we'll go ahead and close up.
Okay, so we use fire as a restoration technique on new native plantings. What is going on with mycorrhizal fungi in these early years? That's a really good question, Pete. I don't think we know a lot about what's going on. One reason why we talk about soil inoculation is because a lot of times we think it takes a while for those, if those spores are gone in those sites, especially in these prairie sites that were once agricultural fields. Um, we think that they might be making their way there, but these AMF spores are large and it's hard for them to travel. So I think that's a great question, and I think we still need to learn a lot about that.
So Alice, would you mind maybe typing your email address there in case people
Sure.
want to follow up with
Yeah,
questions
so Charles, if
afterwards?
you want to send your question to me in an email um, too, or I could try to write you back in an email. There's my email address there.
So agtq4f at
Yes.
mail.missouri.edu.
Hmm. Yep,
Great,
that's it.
and thank you all for participating. We appreciate your support, and we will get this recording online. You'll be able to get it through our website, and then I'll follow up trying to get it posted to YouTube as well. So thank you, Alice.
Okay, thank you, Sherry.
Have a good afternoon,
You
everyone.